Good morning, students. In the last class, we have left this problem of the dihybrid cross, and I have gave you that you may do it at home. Now I hope that you have already completed it, as because it is not a very tough job for you. I again repeating here that in a dihybrid cross we have to consider the two pairs of contrasting character. The first pair is the round wrinkle shape of the seed, and the second pair is the color of the seed, yellow and green. Here the yellow is dominant over green, round is dominant over wrinkle. You will know in the last week you did it. You have seen here on the board. Now after getting the F1 generation where we have got all round yellow seed plant, and when we will find out the possible male and female gametes, then what will happen? We have got 4 different types of female gametes. Suppose it is the female, here is the female gametes and here is the male gametes. I have given here symbol for the male and female, and in this way we will get 4 different types of possible gametes. That is the R, capital Y, capital R, small y, and the third one is the small r. Small r may I write here in this way, small r, capital Y, and the fourth one is the small r and small y. Now these are the 4 different type of gametes. Whether it is the male or female, both will produce similar type of gametes. So here we have put the gametes for female like capital R, Y, capital R, small y, small r, capital Y, small r, small y, and here the same thing has been put. And now you have to cross this one. I did it, this one, and now I would like to do it in this way: capital R, capital Y, small y, capital R, small y, and here capital R, small r. Here is the small y, here is the capital Y. May we write in this way: capital Y, small y, capital Y, small y. Here capital small r and small y. Here is the capital R and small y. Now take this one: small r, capital R, small r, capital Y, capital Y, and after combining this, capital R, small r, and capital Y and small y. The third one is here: small r, and capital Y. Okay. Now this one: small r, and capital Y, small y. Now come to this side: capital R, small r, capital Y and small y. Here capital R, small r, and small y, small y. Here small r, and capital Y, small y. Now come to this side, the last one: small r, small y. In this way we have completed this table, and after knowing the facts of result, then we will get the results of the phenotype ratio for F2 generation. Here it is the gamete for F1 generation, F1 generation gametes, and here is the result for F2 generation. Now look about the result, how you will find out the results. When you will find out the results, then may you write in this way. How you will write here, may you write the first, may you select this first, it is the round yellow. Okay, this is the round yellow, it is 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 not round yellow, it is round, it is not round yellow. Now come to this side, round yellow, it is not round yellow, it is round yellow, it is round yellow, it is not, it is round yellow, this is also no, this is yes. So what is the result now? 1 2 3 4 5 6 7 8 9. So may you write in this way, phenotype ratio. Okay, first you have here seen round yellow, then may you write round yellow, you have given tick mark for this. Now after calculation you have got this one. In this way the first one has completed. Okay, now where we have left one, it is already chosen, it is chosen, now it has left. Now see what is the trait, it is the round and this one is the green. That means now may we write here the second trait with round green, Now see here round green, may you give like this symbol, round green. This is also round green, this is not round green, this is not round green, this is round green, this is not round green, this is not round green. That means we have got round green 3. Okay, now we have completed up to this box. What are the now box left? 1 2 3 4. See what is the result of all these boxes. Here is the wrinkled and this one is the yellow. Here is the wrinkled and this one is the yellow. That means we may write here wrinkled yellow. In our result, we can write in this way, wrinkled yellow. Now we may put again here a sign, like may I have put here a triangle sign, like round yellow. It is also round yellow and it is also round, wrinkle yellow, it is also wrinkle yellow. Okay, so now you have got wrinkle yellow, that is the 3. Okay, wrinkle yellow. Now one box is only here, that is the wrinkle green, only one box is left here, that is the wrinkled green. In this way we have seen that. May you put here sign again. One. That means the phenotypic ratio is here 9:3:3:1. In this way you are able to do the dihybrid cross or experiment. Now students, we have already completed monohybrid. I have also in the last class given you an idea how you worked out the other examples like the purple with white flower for the monohybrid, and here you have got the result of dihybrid cross. Now we have the 3 different laws based on this Mendel experiment. The first 2 laws, law of dominance and the law of segregation, based on the monohybrid cross, and the third law, law of independent assortment, based on the second cross, that is the dihybrid cross. Now look what are these laws. Look here, the different laws of Mendel based on inheritance. Here is the first law, law of dominance. The first law, law of dominance, and the second law, law of segregation, law of segregation. Both laws have been deduced from the monohybrid experiment by Mendel. How it will be deduced? You may look here, law of dominance. According to this law, out of 2 contrasting allelomorphic factors, out of the 2 contrasting allelomorphic factors, only one expressed itself in an individual. The factor which expresses itself is called dominant, while the other, which has not shown its effect, is termed as recessive. So this is the law, first law. According to first law, what we have in case of monohybrid experiment, we have seen that when pure tall and pure dwarf crossed with each other, what will happen in the F1 generation? The tall, only the tall plants obtained, 100% tall plants obtained. That means, although here in this F1 generation, both the factors are the alleles for tallness also and for dwarfness also present, but only one is able to express itself and the other is not effective. That is why it is the law of dominance. This law may have failed in the future when there are some other experiment did on the inheritance of the traits. But so far as our syllabus is concerned, we have to know that this is the first law of Mendel's. Now come to the second law. In the second law of Mendel, what happened? That is the law of segregation. Law of segregation, and it is also known as purity of gametes. I in the last class also told something about this one. Now you write the law and try to understand with the help of the monohybrid experiment. The law of segregation states that when a pair of contrasting factors or allelomorphs genes are brought together in a hybrid, these factors do not blend. These factors, a pair, that is the from this in the hybrid, there are presence of this pair, heterozygote. This is here together in a hybrid. Together here in a hybrid, what are the alleles, what are the factors for the tallness and for the dwarfness? But these factors do not blend, are mixed up. ये दोनों फैक्टर जो है एक दूसरे में मिक्सड अप नहीं हुआ या अपना वजूद नहीं खोया, है ना, are brought in a hybrid. This factor do not blend or mix up, but simply associated for the time being in this generation. इस जनरेशन में ये सिंपली एक दूसरे के साथ है ना. और जब इसको अवसर मिला, जब सेकंड जनरेशन आपने देखा सेपरेट एट द टाइम ऑफ गैमेट फॉर्मेशन, जब गैमेट F1 को सेल्फिंग कराया गया F1 को, तो इसमें से क्या ये हुआ, तो फिर आप देखा इसमें क्या हुआ कि ये दोनों जो है अलग हो गए. तो ये जो है दिस इज़ द सिग्रीगेशन, इसी को कहते हैं सिग्रीगेट करना, अलग होना. और फिर मिलना, दैट इज़ द असॉर्टमेंट, जो हम लोग तीसरा लॉ में देखेंगे। सिग्रीगेट हो जाना और फिर मिल जाना, दैट इज़ नोन एज द असॉर्टमेंट। तो यहाँ पे ये देखिए फिर ये आया। तो इस तरह से आपने देखा होगा F2 में कि आपको इसी के सिग्रीगेट होने के कारण और गैमेट के प्योरिटी होने के कारण फिर हमारा रिजल्ट जो है चेंज हुआ। जबकि F1 में 100% टॉल था, तो F2 में 100% टॉल नहीं हुआ, बल्कि 75% टॉल हुआ और 25% ड्वार्फ हुआ। तो दिस इज ऑल्सो बेस्ड, द सेकंड लॉ इज ऑल्सो बेस्ड ऑन द monohybrid experiment. Now look about the third law, that is known as the law of independent assortment. This is the third law of Mendel's law of independent assortment. Here look carefully in the inheritance of two pairs of contrasting characters. In the dihybrid cross we have seen the characters which are inherited, we have selected from the two pairs, like we are talking about the round and yellow, and in the another pair we have taken wrinkle with green color of seed. Two pairs of contrasting characters. The distribution of factors of each pair in the gametes is independent of the distribution of other pair. जब गैमेट बनने का समय आया तो हमने क्या देखा है ना, कि कोई भी किसी के साथ एसोसिएट हो रहा और फिर किसी के साथ मिल जा रहा फैक्टर, तो इसी का नाम है इंडिपेंडेंट ऑफ असॉर्टमेंट। फिर देखिए इट मीन्स ड्यूरिंग गैमेट फॉर्मेशन सिग्रीगेटिंग पेयर्स ऑफ यूनिट फैक्टर्स असॉर्ट, जो अलग हो गए थे फिर वो असॉर्ट हो सकते हैं। राउंड के साथ येलो आ सकता है, रिंकल के साथ ग्रीन आ सकता है। इस तरह से हम कई माने उसमें 9:3:3:1 के रेशियो से देखें। आप उस एक्सपेरिमेंट यू हैव टू look about that experiment and thereafter you will try to understand the this law. Understand? It means during gamete formation segregating pairs of unit factors, जो यूनिट फैक्टर के पेयर जो अलग हो गए यूनिट फैक्टर्स, यानी कि राउंड अलग हो गया, येलो अलग हो गया, रिंकल अलग हो गया, अलग अलग इसका एलील हुआ, अलग अलग फैक्टर हो गया, और उसके बाद असॉर्ट इंडिपेंडेंट, और फिर एक दूसरे से असॉर्ट किया, मिला इंडिपेंडेंटली। ऐसा कोई शर्त नहीं रहा कि राउंड जो है येलो के साथ ही मिलेगा या रिंकल जो है ग्रीन के साथ ही मिलेगा। इट इज़ नॉट कंडीशनल, it is independent. Understand? The association when we have selected parent, at that time we have associated the more character, like the round shape of the seed associated with the yellow. That means we have selected that type of people and having the characteristic features with round seed and the color of the seed is yellow. But this association, when at the time of formation of gamete they may segregate, अलग हो जाना, अलग हो गया, और उसके बाद फिर जब ये मिला तो ऐसा कोई शर्त नहीं रहा कि राउंड के साथ येलो ही आएगा, रिंकल के साथ ग्रीन ही आएगा। ये एक दूसरे से इंडिपेंडेंट असॉर्ट किया, जिसे आपने टेबल में चेकर बोर्ड में आपने देखा 9:3:3:1 के रेशियो में आपको जो प्राप्त हुआ फिनोटाइपिक रिजल्ट। तो ये था आपका 3 लॉ। अब इसके बाद हम लोग after this you can also do one more exercise depending upon this, that is the dihybrid experiment. Just like in the last class you have seen that one more example is given besides the examples of the dwarf and the tall. Here you may also take consideration like the tall plant with are having purple, tall plant having purple flower, and the second choice dwarf plant having white flower. That means instead of round yellow and wrinkle green in the second experiment. We can do this type of experiment, that is also the dihybrid. You may do it, try to solve the problem. That is the exercise for you. Okay, now we will know about the sex determination in human being. The sex determination, here sex determination, that means what, which factor is responsible to decide whether an offspring is a male or female. Sex determination is concerned with the study of factors which are responsible for making an individual male or female. Whether the individual will male or female, it will be decided by some factors. It has been observed that species use very different strategies. That means there is a no, in all the organism, in all the animals, there is a no any fixed strategy. There is no any specific, like the in case of the human being, when we are talking about the sex determination, that determined by the gene, but it is not true in all cases. Determining the sex of an animal, like there are some examples here. In some animals, sex is determined by environmental factors such as temperature. The temperature at which fertilized eggs are kept, that will decide whether the child will be a male or female. That is here, whether the offspring developing in the eggs would be male or female. Like at high temperature, like 30 to 35 degree Celsius, the females are born. At low temperature, males are born. Here is the one more example from the hen, or the female foal hen will be changed into the male foal due to the influence of the hormone. And that means there is several strategies taken by the organism. May you look here before knowing about the sex determination rules in the human, just one few examples more about the sex determination that governed by the other factors. In regard to the sex determination, we have seen one example in turtle where the temperature will decide. In the case of turtle, we have seen that temperature will be a deciding factor whether a male will produce or a female will produce. Here the second example I have taken from the Bonellia. This is the Annelida. If larva happens to enter the female's body, it develops into male, but if growing alone, it develops into a female. It is the another strategy taken. In the other example here, in the strategy which will taken by the animals as the hormonal control. Due to the hormonal control, the sex determination take place, like in case of the foal hen that will change to the fertile male cock. So this is the way. But in case of the human, the sex determination is determined by the gene. That means the whatever the inherited genes from the parents, that will decide whether a child will be born a female or a male. Let us see what will happen in case of the sex determination in human. Here now we are in this part, that how sex determined in human beings. In human beings, the sex of an individual is largely genetically determined. In other words, the genes inherited from the parents decide whether the progeny would be a boy or a girl. Understand? Now let us see the chromosomal theory of sex determination. Understand? Here look how the sex will be determined in human. In the human body, there are, in human, there are 23 pairs of chromosomes. 23 pairs of chromosomes. Out of these 23 pairs of chromosome, the 22 pairs of chromosomes known as autosomes, and one, this is the 22 pairs of chromosomes known as autosomes, and the second, one pair of chromosome, is known as sex chromosomes. That means the chromosome which will decide the sex. Now, if we will write this one, then we may use like this, autosomes with capital AA. But in case of one pair of chromosome is known as sex chromosome in case of human. This is the two different types, as because male possesses XY. This pair is the one pair is XY, whereas the female possesses XX. Understand? The female possesses XX. Like if we will look about the diagram of the chromosome, like this way. This is XX and this is Y. So that is here in male, the one pair of sex chromosome is known as XY, whereas in female the one pair which can determine the sex in female is known as XX. This is the similar alleles, that means the homozygous, and this is the dissimilar, it is known as the heterozygous. Now how it will decide? Now look here, suppose male and female at the time of gamete formation, what will happen? There are presence of autosomes plus XY. This is the male. In the female, again there are presence of autosomes plus XX allosomes or sex chromosomes. Autosomes and sex chromosomes, that is also known as allosomes. So here the possibilities of, while they will produce gamete, then what the factors, what the genes will inherit it or transmit. Look here, there are two possibilities. In case of the male, the first possibility is the maybe A plus X, okay, This is the first possibilities. The second possibilities may be A plus Y. Understand? But here in case of female, there is only one possibilities, that is A plus X. As because these are the similar. Now during the fertilization, after fertilization what will happen? If this will combine with that, then A plus XX, that means male, sorry, female. And if in this way, then male. The born child will be male. In this way, sex will determined in the human being. That means there are 22 pairs of autosomes and one pair of sex chromosome in male, that is known XY. Similarly, in female, there are presence of 22 pairs of autosomes and one pair of sex chromosome, that is also known as allosomes. Sex chromosomes are autosomes. This is also known as allosomes. So in this way, you have understood about the sex determination. Here you may this structure will help actually all the 23 pairs of human chromosomes and their karyotype, that means what are the shape that have been already found out. That's why I have given here this type of figure to you for your understanding. Okay, now after this, as we have no time to start the new topic like the evaluation, actually मैंने ये सोचा था कि आज ही इसको स्टार्ट कर देंगे, लेकिन नहीं हो सका। तो अब चलते चलते एक और चीज़ जो है मल्टीपल एलील्स के बारे में आपको बता दें, और जिसमें आप दो चीज़ें देखेंगे, मल्टीपल एलील्स देखेंगे और किसी एक कैरेक्टर को डिटरमाइन करने के लिए, और दूसरा आप उसमें कोडोमिनेंस देखेंगे, जिसमें आपको ये लगेगा कि किस तरह से मेंडल्स का लॉ ऑफ डोमिनेंस कहीं कहीं पर फेलियर हो रहा है। एक छोटा सा एग्जांपल आप ब्लड ग्रुप का देखेंगे, उसी से पता चल जाएगा। कार्ल लैंडस्टेनर was the first who discovered the blood groups. He told that blood is not alike in all persons. He has classified blood into 4 different groups: A, blood group A, blood group B, blood group AB, and O. This blood system is controlled by a gene which has 3 different forms, alleles, like that is denoted by the symbols. This symbol is denoted by this symbol, this and small I. So here are 3 different forms of the genes. That is these 3 forms are the alleles of a particular genes that determine the blood group. Here both IA and IB are codominant. That means ये भी dominant और ये भी dominant. अब यहाँ पे देखिए, if you may here consider the law of dominance according to Mendel, then it has been totally failed here, as because there is no 2 factors are going to be dominant. But here the both factors are dominant. Understand? A person carries only 2 of the above 3 genes. 3 genes हैं IA, IB, I, लेकिन एक पर्सन में, किसी एक इंडिविजुअल में, चाहे वो मेल हो चाहे फीमेल हो, इसके 2 फॉर्म ही आएंगे. एक फॉर्म उसका अपने पेरेंट से आएगा, इनहेरिट करेगा. A person carries only 2 of the above 3 gene forms, 1 from each parent. So the blood group of a person depends on which 2 forms of the genes he possesses. अब ये डिसाइड करेगा ब्लड ग्रुप कि उसके पास 2 फॉर्म कौन सा है. अब आइए देखें इसको किस तरह से. बहुत ही ये ईजी है, इसे आप एकदम मिनटों में आप इसको खुद से कर लीजिएगा. देखिए कई पॉसिबिलिटीज़ हैं. नंबर 1 यहाँ पे हम देखते हैं, अगर इंडिविजुअल में ये है, अगर किसी इंडिविजुअल में, इन इंडिविजुअल दिस फॉर्म्स ऑफ द एलील, अंडरस्टैंड, IAA and IA, and also if this form like and I, in both these cases, if an individual inherited from his parent or from her parent this, either this or this, in both cases the blood group will be A. Understand? Now come to know about the others similar on this similar pattern. Like in case of the other, suppose a person and that they getting both IB and once again this IB and I, as it is recessive. This is dominant and this is also dominant. This is dominant, this is dominant, and these 2 both are codominants, and these 2 are dominant over I. That is the 3rd form of the allele. That's why here the blood group will be B. In the 3rd situation, if we look here, the 3rd situation, in the 3rd situation what will happen if IA and IB both, then what will happen? Suppose from one parent inherited IA, from the another parent inherited IB, then what will happen? There is a blood group AB. Understand? And the 4th possibilities is only that how there will be O blood group. If there is a and there is a, that means what they will inherit from both the parent. If an individual will inherit this recessive, which are recessive gene, recessive allele here in the above cases, then there will be possibility of blood group. So in this way we have seen that there are 4 different types of blood group, and this 4 will be how inherited it will be decided by the 3 forms of the genes, that is IA, IB and small I. Understand? In this way we have seen here the multiple alleles, and we have also seen here the codominance.